who's a researcher at the BC Cancer Agency, and she's going to talk to us about abstract 348. Lung adenocarcinoma genomes of never smokers exhibit a greater extent of genomic instability than those of smokers.
Thank you, Dr. Herbst. So my research project has a bit of a different tone than the other panelists up here. This is a preliminary study looking for genomic differences between smoker and never smoker lung tumors. And basically we're trying to find differences that may potentially lead to targeted therapies for one group or the other down the road pending further investigation. Next, please. So we typically think of lung cancer as a disease of smokers, but actually up to 25% of cases arise in people who have never smoked. And if you consider lung cancer in non-smokers as its own disease, it would actually rank seventh as the seventh leading cause of cancer death worldwide. So it's affecting a lot of people, and it definitely demands some attention. So there are some known clinical and molecular features that can distinguish lung tumors that we see in smokers and non-smokers. For example, EGFR mutations are a lot more frequent in never smokers than they are in smokers, and this kind of underlies the reason why we see a higher response to EGFR targeted therapies in the non-smoker group. But not all never smokers have EGFR mutations, so there must be other genetic aberrations that are happening that are driving the different clinical behaviors that we see. Next, please. Thank you. So this is what our group did was basically looked for genomic differences at the DNA copy number level. So these are a type of genetic alteration that can alter the number of genes that you have in your genome. So we often see that if you have an increase in the number of genes or the number of copies of a specific gene, you can activate that gene aberrantly in cancer cells, whereas if you lose copies of that gene, you can inactivate that gene. So this is one mechanism of deregulation that we see in cancer, and this is the results that we've got when we look at the genomes from smokers and never smokers. So we did a comparative study. We had 39 lung cancers from smokers and 30 from never smokers. And this plot is just showing the frequency of alterations throughout the genome. So you can see that they look generally similar, but there are some regions that appear to be different throughout the genome, and there's a few animations if you click it maybe 3 times. So these seem to be preferentially altered in either smokers or non-smokers, and I'll talk about these in a minute. And we also noticed that in the never smoker group, the frequency of alterations seemed to be greater throughout the genome than it was in the smokers. So next, please. Oh, one more. Thank you. So to look at this in a quantitative manner, we calculated the fraction of the genome that was encompassed by DNA copy number alterations for each tumor in our data set, and we compared it between never smokers and smokers. And what we saw was that actually the never smokers have a greater proportion of their genomes altered. And so we think this is evidence that these tumors have a greater extent of genomic instability. And we then went to validate our findings in other data sets, and we saw the same trend in 2 external data sets as well. And next, please. And also, as I pointed out, there seem to be some regions in the genome that were preferentially altered in either smokers or never smokers. And this slide is just showing you some of these regions that we've identified. So one of them is, they're all copy number gains, I should say. And one of them is copy number gain on chromosome 16p. And this was encouraging to see in our study because it kind of validates our approach, because this region has been reported in the literature before. But we also identified regions on chromosome 7p and chromosomes 5q. So we think that the genes in these regions could be important players in lung tumor genesis in never smokers specifically. So altogether, our study, I think we've identified some global or whole genome differences between smokers and non-smokers, as well as the regional differences that we found here. And I think this points to there being different molecular mechanisms of lung cancer development in these 2 groups, which suggests down the road, pending further investigation, that maybe we should be studying and managing lung cancer patients differently based on whether they've smoked or not smoked. Thank you for your attention.
Thank you very much. We now are open for questions or comments. Are you working with other databases now to try to reproduce this?
Definitely, yeah. So we've looked at 2 other data sets so far, but it's definitely a critical thing to do in any study. So there's a lot of publicly available data that we'll be trying to look at to validate this further.
Yes, please.
Susan Mayer from the BMJ. I just wondered, this may be a naive question, but I just wondered if any of the regions that you found of differences had any links with currently known targeted therapies that might lead you forward in looking at different treatment options, and also how this relates to already observed differences clinically in response to therapies.
That's a good question. So the regions that I have on this slide here, they don't contain any genes that I know of that are being used for targeted therapies right now. But I think, if we do some more investigation, we further look at the biological role and how these can play a role in tumor genesis, we may reveal some interesting targets that could potentially down the road be used for the treatment of never smoker lung cancer.
May I add a comment?
Oh, please.
So I think that what the work that this group is doing is like the first step to discover the new genes and then verify what will be the metabolic pathways that are involved. And then we can find the drug that will be the ideal to target that specific pathway. And then we move to the type of study that we are doing that develop this clinically, develop the clinical test, and find a mechanism to enroll the patient. So she has the first step, and then we have the second step illustrated here.
Right, I would just echo that. It might not just be one gene either. It might be a series of genes around a pathway, a nodal point in a tumor. And certainly for those good citizens who don't smoke, we really owe it to them to try to figure out exactly, or who have stopped smoking, how to treat their cancer. We should do it for everyone, but them especially. Okay, next, the next presentation is by